Before we talk about today's lesson on DNA translation, let's review DNA transcription. Pause the video to answer the following questions. What is the job of DNA? The job of DNA is to give instructions to make proteins. Now, proteins are really important for us because they're able to make structures, enzymes, hormones, or even build muscles. Why can't DNA leave the nucleus? We don't want anything to happen to the DNA since it holds valuable information. In order to get this information to the ribosome, we must make a copy of the DNA. This process is called DNA transcription. You can think of the word transcribe as write. So we are writing a copy of DNA in the form of mRNA or messenger RNA. Describe the steps of transcription. First, the two DNA strands must unwind. Then the RNA polymerase matches each DNA nucleotide to an RNA nucleotide to form a messenger RNA or mRNA. Once the mRNA is made, it is sent to the ribosome. The ribosome will read the mRNA to make proteins. Now that we've reviewed DNA transcription, let's move on to DNA translation. If I were to visit a country and didn't know the language, it would be helpful to have a translator by my side. This way, the translator could change my thoughts and ideas from my language to another language. When you think of translate, think change. In our cells, the ribosome must read or decode the mRNA in order to change it into a form that the body could use, which is protein. In order to do this, the ribosome attaches to the mRNA. It will start to read the mRNA when it finds the start codon, AUG. The start codon is like the beginning of a sentence. You wouldn't start reading in the middle of the sentence. You would need to find the first word of the sentence. Once the ribosome finds the start codon, it will match every mRNA codon to a tRNA or transfer RNA codon. The tRNA has a specific amino acid attached to it. In this picture, we see the mRNA, the blue structure which represents the ribosome, and the tRNA. The different colored circles represent different amino acids. When we put all of these amino acids together, we will form the macromolecule protein. The ribosome will continue to read the mRNA and put the amino acids together until it reaches a stop codon. How do you know when to stop reading a sentence? The punctuation mark should signal the end of a sentence. The stop codon is like a punctuation mark. Once the ribosome reaches the stop codon, it will release the protein. Let's take a look at how this looks like in a cell. Here you can see the ribosome attaching to the mRNA. You'll find the mRNA at the top of the screen and the tRNA with the amino acid at the bottom of the screen. The ribosome will start to match each mRNA to a tRNA. And the amino acids will attach to each other. The ribosome will continue this until it finds a stop codon. Once it finds the stop codon, it will release the protein. Now that you've learned how to make a protein through the processes of transcription and translation, let's try an example. There are two strands to DNA. Here is one strand. Find the complementary base pairs of the second strand. Pause the video and write your answers. Remember, for DNA, for every adenine, it matches with thymine, and for every cytosine, it matches with guanine. The first codon of the second strand should be ATG, then AGA, ATA, TGC, TAA. Now transcribe this DNA top strand to an mRNA. Pause the video to write your answers. Remember, the only difference here is for every adenine for DNA, it will be uracil for RNA. Here is the mRNA strand for that DNA. Now that we've transcribed DNA into mRNA, let's translate it so we can make a protein. In order to do that, you need your amino acid chart. We need to use the amino acid chart to figure out the amino acid for each of these codons on the mRNA. The first codon that we'll look at is AUG. When we look at the amino acid chart, we need to find the first letter on the left side of the chart. The second letter will be shown at the top of the chart, and the third letter will be shown at the right section of the chart. Let's take a look at our chart and find AUG to find its appropriate amino acid. The mRNA we're trying to look for is AUG. The first letter of AUG can be found on the left side. Since A is in the third row, I will only focus on the green box. The second letter in AUG is U. I'll find U at the top of the chart. This will narrow down my search. Instead of looking at the green box, I can now focus on the blue box to find AUG. The third letter of AUG is G. I can find this on the right side of the chart. If I move straight across to the blue box, I can find AUG. The amino acid for AUG is methionine. I can abbreviate that as MET. The next codon that we can look for is AGA. The first letter of AGA is A, so I know I need to focus on the third row. The next letter in AGA is G, so I'll take a look at the top of the chart and find G. I can narrow down my search and focus on the blue box. The third letter of AGA is A. By moving across to the box, I can figure out that AGA is arginine. The next mRNA is AUA. So A will be found on the left side, on the third row. The second letter is U. Find it at the top of the chart. So I can focus, instead of that whole entire third row, I can just focus on the blue box. And the third letter is A. So if I move all the way across, I can find that AUA is isoleucine. The next codon is UGC. U is in the first row. G at the top of the chart is in the last column. So I need to focus on the blue box at the last column. The last letter is C. If I move across, I can find out that UGC is represented by the amino acid cysteine. The last codon is UAA. U is found in the first row. A is found in the third column. And the last letter A, if I move it across, will show that it's a stop codon. Here are your amino acids that will combine to make one protein. Try this problem on your own. First, find the complementary DNA base pairs for this top strand. Pause the video to write your answers. Let's transcribe the DNA top strand into an RNA. Pause the video to write your answers. Now translate mRNA into an amino acid by using your amino acid chart. To summarize, we need to transcribe DNA into mRNA. Once mRNA is made, we'll send it to the ribosome. We'll start translation when mRNA and the ribosome attach to each other. The ribosome will read the mRNA to figure out what amino acids must be put together to make a protein.